So picture the Earth in your mind for a second. And I'm talking about, the entire staggering biological tapestry of our planet.
Right. It's a lot to take in.
It really is. I mean, imagine the towering sequoias of the Pacific Northwest just pushing hundreds of feet into the sky, pulling literal tons of mass out of thin air to build their trunks.
Absolutely.
And then, zoom all the way down to the microscopic bacteria that are just thriving in the pitch black, crushing pressure of deep sea hydrothermal vents, the ones feeding off the heat of the Earth's mantle.
Yeah, the extremophiles.
Exactly. And as vastly different as those organisms seem, as completely alien as their environments are from one another, they are all fundamentally woven from the exact same chemical thread.
Which is, of course, carbon.
Right, carbon. Every single known living entity on this planet, from the grass in your yard to the neurons firing in your brain right now as you listen to this, it relies on the intricate, beautiful chemistry of carbon.
It's the structural and functional foundation of everything.
Yeah. It's so absolute, so universally fundamental to our working definition of biology that the entire scientific field of organic chemistry literally just means the study of carbon.
We don't even name it after the element itself.
No, we just call it organic because to us, carbon is life. Period.
It's an incredible monopoly when you really think about it. Carbon is just the undisputed king of the terrestrial periodic table.
Right.
But, as our telescopes peer further out into the dark and as the field of astrobiology expands to look at thousands of newly discovered exoplanets.
Not to mention the crazy exotic environments hiding right here in our own solar system.
Exactly. A really massive universe-shifting question naturally emerges. We have to look at this monopoly and ask, is carbon the only viable blueprint for biology?
Like, is it the only game in town?
Right. Does the periodic table offer other candidates?
Yeah.
Are there other elements capable of supporting the immense chemical complexity, the molecular stability, and, the sheer abundance required to build a living, breathing, reproducing organism entirely from scratch?
And that is exactly the mission of today's edition of Audio Notebook. Because among all the 94 naturally occurring elements, there is one that stands out as the ultimate theoretical alternative.
The astrobiologist white whale.
Yes.
Yeah.
The sci-fi darling itself, silicon. And to give you an idea of the scope of what we're going to unpack today, we are going to completely deconstruct silicon.
From the ground up.
Right, we'll start down at the invisible atomic level to see why it's even considered a candidate in the first place. Then we'll explore the brutal, unforgiving chemical reasons why it completely fails to support life here on Earth.
And it does fail spectacularly.
Oh, it's a disaster. But from there, we'll travel to the extreme, hostile, alien environments in our solar system and beyond where silicon might actually thrive.
And we'll finish up with something that is just mind-bending.
Yeah, looking at new developments in synthetic biology that are actively forcing terrestrial carbon-based cells to use silicon in laboratories right now.
It really is a massive journey. And to understand why we're taking it, we have to start with why scientists even look at silicon in the first place.
Right.
You can think of it in terms of structural similarities. If carbon is the ultimate versatile building block that built absolutely everything around us.
Silicon looks, at least at first glance, like it should fit into the exact same chemical slots.
Exactly.
I always think of it like a cosmic construction kit. If
Yeah.
carbon is, the standard 4-peg Lego brick that you can use to build a house, a car, a spaceship, whatever, and silicon looks like a slightly larger version of that exact same 4-peg brick.
That is a brilliant way to conceptualize it.
But as we're going to see, when you actually try to snap those larger bricks together, the fundamental physics of the universe completely changes the game.
It really does. And to understand why it looks like the same brick, we have to go straight to the periodic table, specifically to group 14.
Right, the vertical call.
Yeah. So carbon sits at atomic number 6. And sitting directly beneath it in group 14 is silicon at atomic number 14. Because they're in the same vertical column, they share a very specific, profound chemical kinship.
Which
They have identical valence electron configurations.
Okay, let's pause there. For those of us who might be, a little rusty on our high school chemistry, valence electrons are the ones on the very outside of the atom, correct?
Exactly.
They're the ones actually bumping into other atoms, doing the interacting, and forming the chemical bonds.
Right. The inner electrons are mostly just taking up space, but the valence electrons are the socialites of the atom.
The socialites, I like that.
Yeah. So carbon has 4 of these valence electrons in its outermost shell. But according to the laws of quantum mechanics, it desperately wants 8 to reach a stable, happy state.
It's trying to fill the shell.
Exactly.
Oh.
We call this the stable octet configuration, which perfectly matches the stable, completely unreactive nature of the noble gas neon. Now, silicon also has 4 valence electrons, and it also desperately wants to borrow or share 4 more to reach the stable octet of its nearest noble gas, which is argon.
So they both have 4, but they want 8.
Yes. And this shared trait, having 4 and wanting 8, is called tetravalency.
Tetravalency, meaning it has 4 active connection points, the 4 pegs on the Lego brick.
Exactly. And tetravalency is the absolute cornerstone of biochemical complexity.
Because it lets you build out in 3D.
Right. Because it has 4 points of connection, carbon doesn't just form simple straight lines. It can form long, sprawling chains, complex branching structures, 3-dimensional rings, and highly intricate lattices.
You need that 4-way bonding capability to build the sheer physical scaffolding that biology requires.
Without tetravalency, you can't build DNA, you can't build proteins, you can't build cell walls.
Okay, so they both have the 4 connection points. They're both the 4-peg Lego brick. So where does the illusion break down? Why aren't they exactly the same?
It breaks down the moment you look at the physical scale of the atoms. Silicon is significantly larger and heavier than carbon.
How much larger?
Well, the specific measurements really matter here. In chemistry, we measure atoms in picometers. And a picometer is 1/trillionth of a meter.
Okay, tiny.
Very tiny. Carbon has a covalent radius of 77 picometers. Its van der Waals radius, which, you can think of as the overall physical personal space the atom claims before it starts repelling other atoms, is 170 picometers.
Got it. 77 and 170.
Right. Silicon, on the other hand, is massively bulkier. Its covalent radius is 111 picometers, and its van der Waals radius is 210 picometers.
So it's taking up vastly more physical space. But why does that matter? If it still have the 4 connection points, shouldn't it still be able to build the same structures, just, scaled up a bit?
That is the intuitive assumption for sure, but the quantum mechanics of atoms don't scale linearly. The reason silicon is larger is because of its quantum shells.
The layers.
Yeah, think of an atom like an onion. Carbon's valence electrons, those 4 socialites we talked about, reside in the second electron shell, or the n=2 shell.
Okay.
Because it's only the second shell out, those outer electrons are physically very close to the center of the atom. Therefore, they are held incredibly tightly by the positive electromagnetic charge of the protons in the nucleus.
They're kept on a tight leash.
Exactly. Silicon's valence electrons, however, are pushed out into a completely new layer, the third electron shell, the n=3 shell.
Because it has more inner electrons caking up the inner shells.
Yes, and this creates a phenomenon called the shielding effect. Silicon's outer electrons aren't just further away, they're heavily shielded by what we call a neon core.
A neon core.
Right, there are 10 inner electrons orbiting between the nucleus and the outer valence shell. Those 10 inner electrons possess a negative charge, which repels the outer electrons and essentially blocks the positive pull of the nucleus.
Oh, wow.
So silicon's outer electrons are weakly bound, far away, and heavily shielded from the core.
I think of it like parenting styles.
Oh!
Yeah, carbon is the ultimate helicopter parent. It keeps its electrons tightly controlled, highly organized, and very close to home. It knows exactly where they are at all times.
That's a great way to look at it.
Right. And then silicon, on the other hand, is like a highly distracted chaperone at a massive, crowded middle school dance. Its outer electrons are way across the gymnasium. They're shielded by a huge crowd of other kids, and they're highly prone to wandering off, reacting unpredictably, or just getting lost in the shuffle.
That is a phenomenal analogy. And it's accurate because that lack of control translates directly to chemical instability. Because carbon is so small and holds its electrons so tightly, its outermost orbitals, specifically the porbitals, which are shaped a bit like dumbbells, can physically overlap with the orbitals of adjacent atoms incredibly efficiently.
They fit together perfectly.
Exactly. This tight, efficient side-by-side overlap is what allows carbon to easily form highly stable double and triple bonds.
And double bonds are non-negotiable for life as we know it, right? Mm-hmm. I mean, the unsaturated fats in our cellular membranes rely on double bonds to stay fluid. The carbon dioxide we exhale relies on double bonds to remain a small, distinct gas molecule.
They are absolutely vital. But silicon's massive size makes this side-by-side orbital overlap terrible. The orbitals are too diffuse, too spread out.
They just can't reach each other properly.
Right. The atoms physically can't get close enough to each other for those orbitals to interlock. In fact, for a very long time, this was actually codified in chemistry textbooks as the double bond rule.
Wait, really? A hard rule?
Yeah, the rule basically stated that heavy elements with a principal quantum number greater than 2, like silicon, simply couldn't form stable multiple bonds at all.
But I remember reading that the rule was eventually broken, right? We did figure out how to force silicon to double bond.
We did, but it took an immense amount of laboratory effort. In 1981, chemists finally managed to synthesize stable molecules with a silicon double bond. They're known as disolines.
Disolines.
But the caveat is massive. Even though we know it is theoretically possible to force it in a highly controlled laboratory setting, multiple bonds in silicon remain highly rare and incredibly reactive.
They don't want to stay together.
Not at all. They want to break apart almost the instant they form. They just absolutely lack the intrinsic, effortless stability of carbon double bonds.
Okay, so we have a bulkier atom with loose electrons that struggles to form complex double bonds. Does this size difference also affect how many other things the atom can physically connect to at the same time? Earlier we talked about tetravalency, the 4 connection points. Does that change?
It does, and this is where silicon's unique geometry comes into play. We call this the coordination number.
Coordination number.
Right. Because carbon is so physically small, it maxes out at a coordination number of 4. When carbon forms 4 bonds, we say it is coordinatively saturated. There physically isn't enough surface area on the atom to crowd more atoms around it.
Full.
Exactly. But silicon is larger. And crucially, because it's in the 3rd quantum shell, it has access to low-lying d orbitals.
And d orbitals are basically just like extra geometric parking spaces for electrons.
That's a great way to put it. They are extra complex orbital shapes that are completely absent in carbon's n equals 2 shell.
So it has more room.
Yes. This means silicon can actually expand its valence shell. It isn't strictly limited to 4 connections. Under the right conditions, it can squeeze in coordination numbers of 5 or even 6.
6 connections.
Yeah, a classic example in chemistry is the hexafluorosilicate ion, where a single silicon atom manages to bond to 6 fluorine atoms simultaneously.
Wait, if we translate that to biology, that completely changes the blueprint. If I'm building a machine and suddenly a gear that is only supposed to connect to 4 things decides to connect to 6, what does that mean for a theoretical silicon organism?
It means we have to rethink the very nature of biological machinery. Earth life relies entirely on enzymes complex carbon-based proteins that act as chemical catalysts.
The workers of the cell.
Right. And they work by having highly specific, rigid, fine-tuned geometric shapes. They operate exactly like a lock and a key. A specific molecule enters the enzyme, it fits perfectly, a reaction happens, and the product is released.
Very precise.
Very. But if you have silicon-based enzymes, you have a completely different dynamic. If a silicon enzyme can suddenly shift from a coordination number of 4 to 5 and then to 6 mid-reaction, you are looking at totally different catalytic mechanisms.
They wouldn't be rigid locks and keys at all.
No, they would be far more flexible, shape-shifting chemical machines. Theoretically, they could process chemicals much faster because they wouldn't require such precise alignment to force a reaction. They could grab molecules in a variety of configurations.
So they would be incredibly high throughput, but perhaps a bit sloppy.
Exactly.
Like a factory line moving at double speed where the workers are just tossing parts together instead of precisely placing them.
That's a perfect visual.
But, okay, if silicon can theoretically form these complex structures, and it can even act as a super flexible, high throughput enzyme, why isn't it in our DNA? I mean, we live on a planet where the crust is literally composed of 27% silicon.
It's everywhere.
It's the second most abundant element on Earth after oxygen. We are swimming in it. Why did the biological history of Earth completely ignore this incredibly abundant resource?
To answer that, we have to look past the atomic structure and confront the brutal thermodynamic realities that make silicon uniquely unsuited for an Earth-like environment.
Energy math.
We have to do the energy math.
And by thermodynamics, you're talking about the raw energy required to build these bonds and the energy required to break them.
Exactly. The absolute foundation of complex biology is something called catenation. Catenation is the ability of an element to build long, stable chains by bonding to itself over and over again.
Like links in a fence.
Yes. And carbon is the undisputed master of catenation. A carbon-carbon bond contains an ultra-robust 348 kilojoules per mole of energy.
Okay, let's ground that number for the listener. What does 348 kilojoules per mole actually mean in terms of physical reality?
Think of bond energy as the physical effort required to break two atoms apart. If you imagine that breaking a carbon-carbon bond takes the physical energy of swinging a heavy steel sledgehammer,
Okay.
then breaking a silicon bond only takes a light, careless tap from a rubber mallet. A silicon bond only contains about 222 kilojoules per mole.
Wow. That is a staggering drop-off in stability.
It is, and it translates directly to thermal fragility. Heat is just kinetic energy, it's atoms vibrating.
Right.
If you try to build a long, complex chain of silicon atoms molecules known as silanes, they are so fundamentally weak that they literally shake themselves apart at normal Earth room temperatures.
Just from the ambient heat.
Yes. The ambient kinetic energy of a warm spring day on Earth provides enough vibration to effortlessly break a silicon polymer to pieces.
So just existing at 70 degrees Fahrenheit is basically like throwing a silicon organism into a molecular wood chipper. Its very DNA would vibrate itself into dust.
Precisely. But the thermodynamic nightmare for silicon goes far beyond just its inability to hold its own hands.
It gets worse.
Much worse. It's not just that its bonds to itself are weak. It's that its bonds to other specific elements create an inescapable thermodynamic trap.
Okay, lay it out for me.
Think about how carbon metabolism works. Carbon forms bonds with hydrogen at about 413 kilojoules per mole, and bonds with oxygen at about 358 kilojoules per mole.
And those values are incredibly close to the 348 kilojoules per mole of the carbon-carbon bond.
Exactly. And that comparability is the secret sauce of life. Metabolism requires molecules to be constantly broken apart, rearranged, and put back together to extract energy.
Right, it's a constant shuffling.
And because the energy costs of carbon bonds are all roughly in the same ballpark, cellular enzymes can easily facilitate this complex, reversible chemistry.
It's like trading currency between countries with roughly the same exchange rate. You can swap dollars for euros and back again without losing all your money in transaction fees. It's a flexible, fluid economy.
That's a perfect analogy. Now look at the silicon economy. Silicon is hopelessly, fatally addicted to oxygen.
Addicted.
Yes. The silicon-oxygen bond is an absolute monster. It contains 452 kilojoules per mole of energy.
Whoa, that is more than twice the strength of the silicon bond.
Exactly. What this means in practical terms is that in any environment where oxygen is freely available, silicon-based molecules will spontaneously, violently, and irreversibly snap into silicon-oxygen structures.
And they won't let go.
Once a silicon atom grabs onto an oxygen atom, it refuses to let go. It falls into a deep thermodynamic pit that biology simply cannot climb out of.
Wait, so if you were to somehow construct a living silicon organism and bring it to Earth, the oxygen in our atmosphere wouldn't just be toxic to it. It would instantly lock its entire fluid molecular structure into a rigid, unreactive state.
It would freeze its metabolism instantly.
Which naturally leads us to what might be the most horrifying biological concept I've encountered in a very long time, the silica problem.
Ah, yes. The concept of exhaling sand.
We have to explain this step by step for the listener because the imagery is so visceral. When we talk about metabolism on Earth, the standard baseline is carbon oxidation.
Right. You eat carbohydrates, which are complex chains of carbon. Your cellular machinery extracts energy from those molecules by breaking them down and oxidizing that carbon.
And the resulting waste product of that chemical reaction is carbon dioxide, CO2.
Yes. And because carbon can form those highly stable, efficient double bonds we talked about earlier, CO2 exists as a tiny, discrete, independent molecule.
It's
a gas at standard atmospheric temperatures, and importantly, it is highly soluble in water. Your cells produce it, it diffuses effortlessly through your cell walls into your bloodstream, travels to your lungs, and you simply exhale it out into the air.
It's elegant. It's a perfectly clean exhaust system. But if a hypothetical silicon organism tries to execute that exact same metabolic process...
It is an absolute catastrophe.
Okay, unpack this for me.
Let's say this organism eats silicon-based compounds and uses oxygen to break them down. It oxidizes silicon. The waste product is silicon dioxide, SiO2.
Okay.
But remember the double bond rule. Silicon struggles immensely to form double bonds with oxygen. Because its orbitals are too large and diffuse, it can't just form a neat little independent SiO2 molecule.
So what does it do instead?
Instead, to satisfy its valence needs, one silicon atom forms strong single bonds with 4 surrounding oxygen atoms. And those oxygen atoms are bonded to other silicon atoms.
Meaning they physically interlock. They
Yeah.
polymerize. And what exactly is a three-dimensional interlocking lattice of silicon dioxide?
It is quartz. It is literal sand.
Unbelievable.
If a hypothetical silicon-based organism were to use oxygen as a metabolic electron acceptor, if it were to breathe oxygen the way you and I do to generate energy, the exhaust product precipitating inside its delicate cellular machinery wouldn't be a neat soluble gas.
It would be solid, jagged, microscopic bricks of quartz crystal. I just want the listener to really internalize that image. You take a breath of oxygen to get energy, and suddenly your lungs, or the interior of your cells, are physically filling up with solid rock.
The sheer mechanical damage to a cell membrane would be devastating.
It would be like trying to run an internal combustion engine where the exhaust pipe pumps out solid gravel.
Exactly.
But hold on, if the bond with oxygen is such an inescapable trap, and breathing it literally produces solid rock, how does an organism even theoretically move past it? Is silicon life basically doomed the second it encounters oxygen?
If it encounters free reactive oxygen, yes, it is absolutely doomed. The basic biological reality is that the physical energy required for the organism to actively excrete grains of solid silica waste from its cells would vastly exceed whatever microscopic chemical energy it gained from the oxidation process in the first place.
It would literally starve to death just trying to take out the trash.
That's exactly right. The only way an organism theoretically survives this trap is by completely, totally removing oxygen from the equation.
Which means Earth is entirely off limits. There's
Yeah.
nowhere on the surface of this planet where it wouldn't encounter oxygen.
No.
But it's not just the oxygen in our air that's the enemy, right? It's the water in our oceans.
Yes, this is what we call the aqueous nightmare.
The aqueous nightmare.
Water is universally considered the ultimate solvent of terrestrial life. We are essentially walking bags of highly organized, carefully compartmentalized water. Water facilitates everything we do.
Right.
But for complex silicon compounds, things like long-chain silanes or siloxanes, water is an aggressively corrosive acid,
It destroys them.
instantly and violently. It's a chemical process called rapid hydrolysis. If you drop a complex organosilicon molecule into liquid water, the water molecules essentially attack it.
How so?
The oxygen atom in the water molecule has lone pairs of electrons that are highly attracted to the positively charged silicon atom. It attacks the silicon, cleaving the bonds, breaking the complex molecule down into completely inert, insoluble silicates.
And it produces a byproduct, right?
Yes, and in the process, releasing highly explosive hydrogen gas.
Wow. So to summarize the terrestrial situation, Earth has warm temperatures that physically shake weak silicon bonds apart. It has an oxygen-rich atmosphere that turns its metabolism into solid rock. And it has sprawling oceans of water that act like boiling acid, actively dissolving its cellular machinery and generating explosive gas.
Exactly. Earth is, objectively and comprehensively, a toxic hellscape for silicon chemistry.
Which is an incredible perspective shift. We always view Earth as this perfect, nurturing cradle of life, but to a silicon organism, this is the most terrifying planet imaginable.
It really is.
But having established that Earth's water and oxygen are an absolute death sentence, the conversation logically has to move off-world. If we want to find silicon life, we have to scour the universe to find the exact, bizarre environmental niches where all of silicon's catastrophic weaknesses suddenly invert and become its superpowers.
And this raises a fascinating question for astrobiologists. What does a biologically active, silicon-friendly planet actually look like?
Right. Where do we even start?
To answer that, we usually start by looking at high-temperature havens.
Because, as you mentioned, carbon biology is thermally fragile. We burn.
We burn very easily. Complex carbon-based proteins, DNA strands, the delicate lipid bilayers of cellular membranes, they all begin to denature, char, and essentially melt at around 400 Kelvin, which is roughly 127 degrees Celsius.
That's just above boiling water.
Right. Water boils away, proteins irrevocably unfold, and carbon life unequivocally ends. But silicon? Those exact same silicon-oxygen bonds that are a deadly trap on Earth are practically fireproof.
They can take immense heat.
Incredibly high temperatures. A molecular backbone built of silicon oxygen or silicon aluminum can maintain perfect structural integrity at temperatures that would instantly vaporize organic carbon matter.
Okay, so where do we find environments like that?
Astrobiologists look specifically at high-temperature niches. Think of Jupiter's moon Io, which is the most geologically active hypervolcanic body in our entire solar system.
Just covered in volcanoes.
Covered. Or think of hot Jupiter exoplanets in incredibly tight, hugging orbits around their host stars, where the surface is literal, churning molten rock.
So in a place like that, you obviously wouldn't have soft, squishy, water-filled cells. What does life look like in a volcano?
In those environments, we hypothesize life based entirely on molten silicate chemistry. We're talking about crystalline entities, often theorized as magmobes.
Magmos, I love that term.
It's a great term. These are hypothetical organisms living happily submerged in lakes of liquid magma, utilizing the immense ambient thermal energy to drive their highly stable, fireproof metabolic processes.
It sounds like something straight out of a science fiction novel.
Yeah.
But it's fundamentally grounded in the actual mathematically proven thermodynamic limits of the element.
Exactly.
But if high heat is one avenue, what happens if we flip the thermostat in the complete opposite direction? Out to the extreme cold of the outer solar system. Things get equally weird there, don't they?
They do. This is the cryogenic niche, and it solves a massive problem for silicon.
How so?
If you drop the environmental temperature below 100 Kelvin, something fascinating happens to carbon. It essentially freezes solid.
It's too cold for it to do anything.
Right. The activation energy required to make carbon atoms react and do the dynamic work of cellular metabolism is simply too high when there is so little ambient kinetic energy. Carbon chemistry grinds to a complete, irreversible halt.
It's like an engine whose oil has frozen solid. The parts are there, but they absolutely refuse to move.
Exactly. But remember earlier how we said silicon's bonds are weak? That silicon bonds just shake apart from the ambient heat of a warm day on Earth?
Right, the thermal fragility.
In a cryogenic environment, that specific weakness becomes an absolute vital asset.
Really?
Because the bonds are inherently weaker, they require significantly lower activation energy to break and reform. So, in the extreme cold, where carbon is completely inert and useless, silicon's fragile bonds actually allow a sort of slow-motion biology to tick forward.
Oh, wow.
The bonds are weak enough to be manipulated by the tiny amounts of energy available.
And when we talk about cryogenic environments, there is one place in our solar system that completely steals the astrobiological spotlight.
Saturn's largest moon, Titan.
Titan, the methane world. If you are imagining landing on Titan right now, you might think you're just looking at a dead, frozen wasteland, but it's actually one of the most dynamic bodies we've ever observed.
Titan is truly extraordinary. It is the only moon in our solar system with a dense, substantial atmosphere, mostly composed of nitrogen, much like Earth's.
Right.
But what makes it unique is that it has a fully functioning liquid weather cycle that parallels Earth's water cycle. There are dense clouds, it rains, and there are vast flowing rivers and massive surface lakes.
But it's not raining water.
No. The average surface temperature is a bone-chilling 94 Kelvin, which is negative 179 degrees Celsius.
That is unimaginably cold.
At that temperature, all the water on Titan is frozen as hard as terrestrial granite. It literally forms the actual bedrock of the moon.
So the water isn't a liquid solvent anymore.
Exactly. So the threat of rapid hydrolysis, that aqueous nightmare we discussed, where water dissolves silicon structures, is completely eliminated because the water is solid rock.
So what are the lakes made of?
The lakes and the rain on Titan are composed of liquid hydrocarbons, primarily liquid methane and ethane.
And how does silicon fare in a lake of liquid methane at negative 179 degrees?
It thrives. Complex silicon molecules, like those fragile silanes and polysilanes, are perfectly stable in liquid methane. They don't break down, they don't explode.
They just sit there.
They can persist, interact, and potentially form the vast complex polymers that act as the precursors to an alternative biochemistry.
And we are actually sending a mission there to check this, right? NASA is building a probe.
Yes, the Dragonfly mission, currently scheduled to arrive at Titan in 2034. It is an awe-inspiring concept. It's essentially a car-sized, nuclear-powered rotorcraft that is going to fly through the thick Titan atmosphere, hopping from location to location across the surface.
A flying nuclear drone exploring an alien moon. I mean, the engineering alone is staggering. But how is it going to look for silicon?
Dragonfly's primary mission is technically to study the prebiotic chemistry of carbon, the complex organic chemistry before biology that we know exists on Titan. But it carries an incredibly powerful instrument suite.
Tell me about it.
Specifically, the DRAG-NS instrument, which stands for Gamma Ray and Neutron Spectrometer.
How does a gamma ray neutron spectrometer work? That sounds incredibly complex.
Well, it's actually a brilliant piece of physics. The instrument fires a pulse of high-energy neutrons directly into the dirt beneath the lander.
Okay.
When those neutrons strike the atoms in the soil, the atoms get excited and emit gamma rays. Crucially, every different element emits a gamma ray with a very specific, unique energy signature.
Like a fingerprint.
Exactly like a highly specific elemental fingerprint. Dragoness reads those gamma rays, allowing it to measure the bulk elemental abundances of the surface materials without ever needing to dig or process a physical sample.
So it essentially reads the chemical barcode of the ground. It can tell us exactly what elements are sitting there.
Exactly. So for the very first time in human history, we will get direct ground truth data on whether silicon is biologically available on the surface of Titan or if it's entirely locked away deep in the moon's unreachable core.
Wait, I have to stop you there. Because while Titan sounds like a perfect safe haven, there's a massive chemical catch, isn't there? We talked about this idea of a solvent. If liquid methane is so incredibly cold and chemically it's so weak, how does it dissolve the massive long-chain polymers needed for genetics?
Right, that is the issue.
I mean, if you think of what we call the solubility paradox, chitin has all the parts for a silicon engine, but isn't it missing the oil to make them actually move and fold together?
That is the exact paradox, and it is the primary overarching obstacle for the entire liquid methane hypothesis. Methane is what we call a nonpolar solvent.
Meaning it doesn't have a charge?
It completely lacks the electrical charge imbalances that make water such a powerful solvent on Earth. Because it's nonpolar, it's great at protecting the silicon bonds from being destroyed, but it's terrible at dissolving large, complex, heavily structured molecules.
So what happens to the molecules?
If your genetic code gets too long or too complex, the methane simply can't hold it in solution. The molecule just precipitates out and sinks to the bottom of the lake as a lifeless, inert sludge. It can't fold into the complex 3D shapes required to act as an enzyme.
So where in the universe do we find a liquid solvent that perfectly balances these contradictory needs? Something that protects silicon from the destructive hydrolysis of water, but is chemically powerful enough to dissolve, manipulate, and fold massively complex polymers.
To find that, we actually have to look much closer to the sun. We have to look at Venus.
Venus. But the surface of Venus is a literal inferno. It's hot enough to melt lead. It's 737 Kelvin down there. The atmospheric pressure would crush a submarine.
The surface, yes, is profoundly hostile. But if you ascend 50 to 60 kilometers up into the thick Venusian atmosphere, you reach the continuous cloud layers.
Okay, up in the clouds.
Up there, the pressure and temperature are actually remarkably temperate, surprisingly Earth-like. But those massive global clouds are not made of water vapor. They are made of concentrated sulfuric acid.
Concentrated battery acid.
Yes. And for a very long time, the scientific community assumed that an environment composed of concentrated sulfuric acid must be entirely sterile, completely devoid of complex chemistry.
I mean, that makes sense. Acid destroys things.
But in 2020, brilliant research published by Petkowski and his colleagues completely upended that assumption. They conducted a massive deep dive into the underlying chemical properties of sulfuric acid as a potential solvent, specifically for complex organosilicon molecules.
And what did their research actually reveal?
They found that concentrated sulfuric acid actually acts as a brilliant, highly effective solvent for complex silicon chemistry.
Wait, really? It doesn't destroy it?
Unlike water, it does not trigger rapid hydrolysis. It doesn't explosively destroy the molecules. In fact, it actively stabilizes a vast diversity of highly complex silicon structures far better than water ever could.
That is wild.
It has the immense solvation power, the chemical muscle, so to speak, needed to physically force massive polymers to fold into precise geometric shapes, much like water does for terrestrial proteins, but it is uniquely, surprisingly compatible with the fragile silicon backbone.
So, if I'm understanding this correctly, the temperate clouds of Venus could theoretically be teeming with microscopic silicon-based life, utilizing concentrated sulfuric acid exactly the way we use water.
Theoretically, yes. If we look at the overarching alternative solvent scorecard, it is ultimately a game of distinct chemical trade-offs.
Let's run through the scorecard.
You have liquid methane at 91 to 112 Kelvin. It successfully protects silicon but aggressively suffers the solubility paradox.
Right, the sludge at the bottom of the lake.
Yeah. Then you have liquid ammonia between 195 and 240 Kelvin. It dissolves things incredibly well, but ammonia has a lone pair of electrons on its nitrogen atom.
Does that mean it attacks the silicon?
Yes, this makes it prone to causing what we call nucleophilic attacks, where the ammonia aggressively attacks the positively charged center of the silicon atom, cleaving the structures apart almost as violently as water does.
Ouch. Okay. What else?
Then you have exotic solvents like formamide, which works beautifully across a huge temperature range, but it's cosmically rare in large ocean-sized quantities.
Now, who wins the scorecard if you're placing a bet on where the chemistry actually works?
Based on current models, concentrated sulfuric acid, like we see on Venus, and supercritical liquid carbon dioxide, which behaves like a cross between a gas and a liquid at extremely high pressures, are increasingly viewed by astrobiologists as the absolute best candidates for driving the complex, weird chemistry folding of alien proteins.
Wow. We've looked deep into space, from the freezing methane lakes of Titan to the acid clouds of Venus. But I want to completely pivot the conversation now.
Okay, let's pivot.
Because the most shocking evidence for the viability of silicon life doesn't come from a telescope.
Wow.
And it doesn't come from a deep space probe. It comes from a laboratory in California.
Ah, yes.
I want to transition from theoretical astrophysics straight into synthetic biology. Because human ingenuity is actively, forcibly bridging the carbon-silicon divide as we speak.
And this is where the history of biology really gets rewritten. Because historically, over 4 billion years of evolution, not a single known organism on Earth had ever forged a natural carbon-silicon bond.
Wait, not one. I thought diatoms, those little microscopic algae that make up a huge part of the ocean's plankton, use silica.
Diatoms do use silica, but strictly structurally. They pull dissolved silicon dioxide out of the seawater to build incredibly intricate, beautiful, microscopic glass shells around themselves for protection.
Just as armor?
Exactly. But they do not integrate that silicon into their actual internal biochemistry. They never forge bonds between carbon and silicon inside their proteins, their enzymes, or their DNA.
Not a single carbon-silicon bond.
Never. For decades, this absolute profound absence was held up by chemists as definitive proof that biology was fundamentally structurally allergic to silicon.
Until 2016.
Until 2016, a landmark study came out of Caltech led by Frances Arnold, who, it's worth noting, actually won the Nobel Prize in Chemistry for pioneering the underlying technique they used.
Okay, what did her team do?
Her team decided to see if they could coax nature into doing what it had seemingly never done in 4 billion years of Earth's history.
But how do you force a cell to use an alien element? You can't just inject silicon into a cell and ask it nicely to build a new protein.
You use a revolutionary technique called directed evolution. They didn't try to build a new organism from scratch. Instead, they scoured nature for a starting point.
What were they looking for?
They looked for an enzyme that already dealt with challenging chemical environments and found a protein called cytochrome C. Specifically, they isolated cytochrome C from an extremophile bacterium called Rhodothermus marinus.
And where does that bacteria naturally live?
In boiling, highly extreme, mineral-rich hot springs in Iceland.
Boiling hot springs.
Yeah. Because this organism survives in such extreme heat, its proteins are inherently incredibly robust and structurally stable. When Arnold's team examined the specific cytochrome C protein, they found what they call a promiscuous latent ability.
Promiscuous. In a biochemical sense, what does that mean?
It means the enzyme isn't perfectly rigidly strict about what it interacts with. While its natural jibe has nothing to do with silicon, they discovered that under the right, highly specific artificial laboratory conditions, this carbon-based enzyme could actually be tricked into catalyzing the formation of organosilicon molecules.
It actually did it.
It could physically forge a true carbon-silicon bond, but only at incredibly trace, barely detectable levels. It was highly inefficient.
But it was a proof of concept. The door was cracked open a millimeter.
Exactly. So they used directed evolution to push that door wide open. Directed evolution essentially mimics Darwinian natural selection, but hyper-accelerated.
How does it work in practice?
They took the physical DNA sequence coding for the cytochrome C protein and intentionally introduced random genetic mutations. They then inserted this mutated DNA into generations of bacteria, grew them in vats, and screened them.
How do you screen them? Like, what are you looking for?
You test the entire batch to see which individual bacteria are slightly better at forging that carbon-silicon bond. Most mutations do nothing or break the enzyme completely, but a few mutations slightly improve the enzyme's new ability.
Okay.
They found the winners, extracted their DNA, introduced a new round of random mutations, and repeated the entire process.
You are literally breeding them for an affinity to silicon.
Yes. And the results of this selective breeding were absolutely staggering. By mutating the DNA over just 3 generations, they engineered an entirely new enzyme that was 15 times more efficient at creating organosilicon molecules than the absolute best industrial synthetic chemical catalysts designed by human engineers.
Wait, 3 generations. We usually think of evolution taking millions of years of blind, grinding luck. If nature can figure out how to master a completely alien chemistry in 3 generations in a laboratory vat, what could a biological system do with a billion years of selective pressure on a weird, extreme exoplanet?
That is the exact profound question astrobiologists are asking now. The implications of this 2016 Caltech breakthrough are massive, and we can broadly break them down into 3 key scientific points.
Lay them out for us.
First, metabolic compatibility. The study definitively proved that complex biological cellular machinery can handle fragile carbon-silicon bonds perfectly, right inside a living cell at a neutral pH and room temperature, without the protein breaking down or the cell dying of heavy metal poisoning.
It totally shattered the long-held idea that silicon is inherently toxic to life's machinery.
Precisely. Second is evolutionary speed. As you just noted, it proved that the mechanics of nature are not rigid. When pressed by selective pressures, biological systems can adapt to utilize non-terrestrial elements terrifyingly fast. The genetic architecture is far more plastic than we assumed.
And the third implication, and let me guess, this has practical applications for us right now.
It does. The third is sustainable synthesis. This has immediate, massively profitable applications for humanity today. Organosilicon molecules are absolutely essential for modern human life. They are required to manufacture advanced pharmaceuticals, agricultural chemicals, and the semiconductors in our electronics.
And how do we usually make them?
Historically, making them industrially requires incredibly toxic, hazardous chemical solvents, extreme heat, and vastly expensive precious metals like iridium to act as catalysts. But Arnold's biocatalysts can now manufacture what we call enantiopure organosilicon at room temperature in plain water using renewable bacterial vats.
Wait, enantiopure is a great word, but let's break that down. That relates to the handedness of molecules, right? Like right-handed and left-handed versions of the exact same chemical.
Yes. Many complex molecules have a mirrored version of themselves, a left-hand and right-hand version called enantiomers. In pharmaceuticals, this is crucial. Sometimes the right-handed version of a drug cures the disease, but the left-handed version is highly toxic.
Oh, wow.
Industrial chemistry often creates a messy 50-50 mix of both, which is hard to clean up. But biological enzymes are incredibly precise. They produce enantiopure molecules, meaning they only create the exact left or right-handed version you want with zero toxic byproducts.
It's an absolute revolution in green chemistry. We are using biology to manufacture our industrial silicon compounds. But, biology isn't just about building things up. Metabolism is a two-way street. It's about breaking things down for energy, too.
Which brings us to the highly significant 2024 update. Arnold's team didn't just stop at forcing biology to build the bonds. Recently, they successfully engineered completely new enzymes that can actually break the silicon-carbon bond.
Which means they established that a full metabolic cycle is biochemically possible.
Exactly. For a true organism to survive, it must be able to synthesize molecules for growth, but it equally needs to break those molecules back down to extract energy and recycle the raw materials. By proving that biology can be rapidly engineered to do both sides of the equation, they essentially proved that a complete, closed-loop biological metabolism involving silicon is chemically viable.
Okay, so since we know that biological machinery can physically manipulate silicon, and we know there are extreme environments in the universe where it theoretically won't spontaneously combust or turn to solid sand, let's play Dr. Frankenstein for a minute.
Let's do it.
Let's theorize what a purely silicon-based organism actually looks like under a theoretical microscope.
Yeah.
How does it manage the two fundamental requirements of life? How does it store its genetic data? And how does it physically eat?
This takes us out of the lab and into the fascinating realm of theoretical genetics and extreme metabolism. Let's start with genetics. Long-term, stable information storage is non-negotiable for life to exist and evolve.
Right. Carbon life uses the famous double helix of DNA, and the rungs of that ladder are held together by a highly stable sugar phosphate backbone.
Exactly, but you can't just magically swap the carbon atoms in our DNA for silicon atoms. The geometry is wrong, the atoms are too bulky, and the bond strengths are far too weak.
The ladder would simply fall apart.
Yes. Silicon requires structural stability through what we call heteroatoms, meaning you have to mix silicon with other alternating elements to form a strong, stable backbone.
So what are the leading theoretical candidates for a silicon genetic code?
Astrobiologists focus primarily on two leading candidates, polysiloxanes and polysiloxanes.
Let's start with polysiloxanes.
Polysiloxanes are what we commonly call silicones in everyday life. Structurally, they're built on a constantly alternating backbone of silicon-oxygen-silicon atoms.
Wait, hold on. Didn't you just say earlier that the silicon-oxygen bond was the deadly trap? The whole breathing sand problem where it turns into rigid quartz?
On Earth, yes, it's a metabolic dead end because you can't easily break that bond to extract energy. But if you are using it for your genetic blueprint and you are in a highly extreme corrosive environment, say the concentrated sulfuric acid clouds of Venus, that extreme unbreakable stability becomes exactly what you need to protect your genetic code from being dissolved by the acid.
So the trap becomes the shield.
Exactly. Polysiloxanes are incredibly interesting because they are highly flexible, wildly stable, and can easily loop back on themselves to form complex rings. They are a perfect theoretical scaffold for genetics in a high temperature or highly acidic environment.
That makes perfect sense. You want your DNA to be the toughest part of the cell. What about the other candidate, the polysiloxanes?
Polysiloxanes use an alternating backbone of silicon, nitrogen, silicon. Now, these structures are ferociously reactive in water. They would be violently destroyed instantly on Earth.
But off Earth.
If you place them in a cryogenic environment, like the liquid ammonia oceans that are theorized to exist beneath the crust of some icy moons, their reactivity completely changes. In that extreme cold, they possess exactly the right level of chemical reactivity necessary to facilitate the complex, continuous read-write functions of information storage.
Okay, so they have a genetic blueprint. The software is intact. But a blueprint doesn't do anything without a power source. How does a silicon organism actually eat? How does it generate energy?
We have a few robust theoretical models for alternative metabolic energy. The first is what we call the selenium-silicate cycle.
Yeah.
Imagine a high-pressure, incredibly hydrogen-rich environment, perhaps deep down in the dense, crushing crusts of gas giants like Jupiter or Saturn. Deep in that high-pressure zone, an organism could essentially breathe by chemically reacting volatile selenes, those silicon-hydrogen molecules, with heavier elements like selenium or tellurium.
Reacting it with selenium to release stored energy.
Yes. The chemical reaction releases a flow of electrons that the cell can harness. And this isn't purely wild speculation. It perfectly mirrors how certain extremophile anaerobic bacteria right here on Earth breathe sulfur compounds instead of oxygen to survive deep underground.
But what about the magmobes? The theoretical organisms we talked about living in literal lakes of churning lava on a moon like Io. How does an organism submerged in a volcano eat? There are no distinct gases to breathe.
This is where we have to completely redefine our concept of metabolism. In a highly turbulent lake of liquid magma, chemical gradients might not be the most reliable or accessible energy source. Instead, the expert consensus points to two incredibly wild physical possibilities: mechanotrophy and radiotrophy.
Mechanotrophy, meaning eating, movement, consuming kinetic energy.
Essentially, yes. Mechanotrophy involves utilizing the immense physical kinetic energy of the rapidly flowing, convecting liquid rock. The organism would anchor itself to a solid substrate and deploy complex rotary motor proteins into the current.
Like little turbines.
You can literally picture them as tiny biological turbines. As the heavy, viscous magma currents flow past, they physically spin the protein. That physical spinning motion creates torque, which the cell uses to directly force molecules together, generating the chemical energy the cell needs to survive.
That is mind-bending. So instead of breaking down the chemical sandwich for energy, this thing operates like a microscopic water wheel anchored in a roaring river of lava. It just lets the physical brute force current power its cellular machinery.
It truly does. We actually have an analogy in our own biology, ATP synthase. The enzyme that generates energy in our cells literally operates as a spinning rotary motor. Mechanotrophy just scales that concept up to harvest environmental currents.
And what was the other one? Radiotrophy.
The other option, radiotrophy, is equally alien. Highly volcanic environments are often heavily laden with radioactive isotopes. A radiotrophic magnobe wouldn't eat physical food at all. It would harvest highly energetic gamma radiation directly from the environment.
Just absorbing radiation.
It would likely use heavily shielded, highly complex melanin-like pigments to safely capture the destructive radiation and safely convert that radiation into metabolic energy.
Harvesting gamma rays for breakfast. That is staggering. But I have to imagine that operating at those extreme energy thresholds, whether you are harvesting sluggish methane chemistry, lava currents, or ambient gamma rays, completely changes the pace of life.
Drastically. In these extreme environments, particularly the cryogenic ones like Titan, where molecular movement is incredibly slow, Chemical reactions are profoundly sluggish.
Everything just creeps along.
The pace of life would be drawn out to scales we can barely comprehend. A single microscopic cellular organism might require decades or even centuries just to gather enough ambient energy to complete a single basic reproductive cycle.
Okay, so we know what the chemistry looks like. We know the extreme hostile environments it needs to survive, and we can theoretically model its genetics and its utterly bizarre metabolism. But here is the multi-billion-dollar question. How do we actually spot it? If we're pointing a telescope at a planet 20 light years away, how do we definitively prove that a magmobe or a selene-breathing cell is actively operating down there?
That is the absolute bleeding edge frontier of modern astronomy. And the very first rule when developing these detection strategies is that the astrobiological community must actively acknowledge and actively avoid carbon chauvinism.
Carbon chauvinism, that's a fantastic term.
It precisely describes the historical, deeply ingrained bias in our search for extraterrestrial life. Traditionally, when astronomers point a telescope at an exoplanet, they scan the planet's atmosphere looking for familiar, comforting gases. Oxygen, ozone, massive amounts of methane.
The things that make Earth look like Earth.
Right. They look for the specific exhaust gases produced by terrestrial carbon-based biology. But if you are explicitly looking for a completely alien, silicon-based biosphere, looking for Earth gases, absolutely blinds you to reality.
You just miss it entirely.
You will look right at a thriving, incredibly dense alien biosphere and mathematically classify it as a dead rock simply because it doesn't have an oxygen atmosphere.
So what is the alternative strategy? How do you look for life without looking for the chemicals of life?
The alternative is searching for what we call agnostic biosignatures. Instead of looking for specific familiar chemical compounds, we need to look for mathematical patterns of biological activity. We're looking for the energetic fingerprint of life itself, regardless of the elemental ink it happens to be using.
And how do we actually do that from light years away?
There are three primary, highly sophisticated detection strategies currently being developed and deployed. The first is analyzing molecular complexity, often utilizing advanced mathematical tools like the life tracer algorithm.
I want to understand this. How does an algorithm like Life Tracer actually differentiate between a rock and a living organism?
It analyzes the structural complexity and distribution of a chemical sample. Think of abiotic geochemistry, just rocks, ambient heat, radiation, and time. Abiotic processes produce a highly predictable, relatively random, and mathematically simple distribution of molecules.
Okay, that makes sense.
Biology, however, is a highly optimized factory. It produces highly organized, incredibly complex, specific suites of molecules in quantities that simply defy basic statistical probability. Life Tracer mathematically maps this complexity to prove beyond a shadow of a doubt that a guided biological engine must be manufacturing them.
You know, I think of it like the classic watchmaker analogy. It's like walking along a beach and finding a fully assembled, ticking mechanical pocket watch in the sand.
Right.
You don't need to actually see the watchmaker. You don't even need to know what the watch is made of. The sheer, organized complexity of the gears mathematically proves that the wind and the waves didn't randomly assemble it. Life Tracer is just looking for the chemical equivalent of those gears.
That is a perfect summation. The complexity itself is the undeniable signature of a biological process.
Okay, but complexity alone doesn't prove it's alive today. It could just be fossilized chemistry from millions of years ago. Is there a way to look at a planet and see if the engine is actively running right now? What's the second strategy?
The second strategy absolutely addresses that. It's called chemical disequilibrium. This involves analyzing an atmosphere and finding a state that is deeply, deeply out of thermodynamic balance.
What does that look like?
We look for volatile gases coexisting that logically, thermodynamically, should have reacted with each other and completely disappeared millions of years ago.
Like finding a lit, burning match in a room completely full of oxygen. If it's still burning, something must be actively, continuously striking new matches.
Precisely. If we scan a distant exoplanet and find highly volatile selene gases perfectly mixed into an atmosphere that is deeply dominated by heavy oxides, that screams active metabolism.
Because they shouldn't exist together.
Based on thermodynamics, those volatile gases should instantly react and fall to the ground as inert sand. If they are persistently, continuously present in the atmosphere, some vast, active biological process is continuously pumping them out faster than they can be destroyed.
That's brilliant. And the third detection method?
The third is isotopic fractionation. Elements naturally come in slightly different weights, known as isotopes. Silicon, for example, comes primarily as the lighter silicon-28 and the
Yeah.
very slightly heavier silicon-29.
Okay, light and heavy versions.
Right. Now, the fundamental rule of biology across the board is that life is inherently lazy.
Highly relatable.
It's universally true. Biological enzymes always prefer to pull in and process the lighter isotopes because they require fractionally less kinetic energy to metabolize. It's the path of least resistance.
So it sorts the atom.
Yes. Over millions of years of continuous biological activity, a massive biosphere will dramatically mathematically skew the natural isotopic ratio of its entire environment. If we point a telescope at an exoplanet's atmosphere and see a massive, completely unnatural abundance of lighter silicon-28 compared to heavier silicon-29, that isotopic fractionation is a massive, glowing red flag for planetary scale biological processing.
And the incredible thing is that with the new generation of space telescopes, we are actually starting to get data precise enough to see this kind of stuff now.
We absolutely are. The James Webb Space Telescope, or JWST, is already returning completely mind-blowing discoveries that are reshaping astrobiology. In late 2024 and early 2025, JWST turned its massive mirrors toward a few highly intriguing exoplanets, specifically WACE SP-107b and VHS 1256b.
And how exactly does it look at them?
It uses a technique called transit spectroscopy. It essentially stares at the host star. When the exoplanet passes in front of the starlight filters through the very edge of the exoplanet's atmosphere.
So it's backlit.
Yes. Different chemical molecules in that atmosphere absorb very specific, exact wavelengths of light. By analyzing the light that finally reaches JWST, astronomers can see a barcode of missing light, which tells them exactly what molecules the starlight passed through.
And when they read the barcode for those specific planets, what did they see?
They definitively detected clouds, but absolutely not clouds of water vapor. They detected vast, swirling, dynamic atmospheric clouds made entirely of silicates. Sand clouds.
That is wild. Are we openly suggesting that those silicate clouds are biological in origin?
We have to be careful. Currently, the standard astrophysical model classifies these specific worlds as abiotic popcorn planets.
Popcorn planets.
The working theory is that extreme atmospheric microphysics cause silicate particles to physically condense, settle lower into the atmosphere, violently mix due to heat, and pop back up like popcorn in a hot pan.
Okay, so it might just be weather.
Right. But the true significance here isn't that we definitively found silicon life. It's that JWST unequivocally proved that we have the technological capability to resolve and study the silicon cycle on alien worlds right now.
We can literally watch the silicon move through the atmosphere of a planet light years away.
Yes. Resolving the physical presence of those clouds is step one. Step two, which future observation missions will fiercely tackle, is applying those agnostic biosignatures we just talked about. The molecular complexity, the active chemical disequilibrium, the isotopic ratios to those exact silicate clouds to see if the chemistry is purely random, abiotic geology, or if biological engines are actively interfering with the cycle.
This has been absolutely fascinating, but we are bringing this edition of Audio Notebook to a close here. But to finish off, we have to zoom out one last time, because despite all this incredible math, the evolutionary breakthroughs in the lab, and the stunning new telescope data, there are massive, unresolved paradoxes hanging over the entire field of silicon astrobiology. Paradoxes that keep scientists up late at night.
Yes, the vast open questions. And the most significant, daunting theoretical hurdle currently facing astrobiologists is what we call the chirality paradox.
Chimality. We touched on this briefly when we talked about enantiopure drugs, but let's dive deeper. This means handedness, right? Like the fundamental difference between your left and right hand.
Exactly. Your hands are mirror images of each other, but they cannot be perfectly superimposed. Molecules work the exact same way. And Earth biology is incredibly strict about this.
Give me an example.
Every single amino acid used to build proteins in carbon life on Earth is strictly left-handed, or the L form. Every single sugar used in the backbone of our DNA is strictly right-handed, or the D form. We call this strict uniformity homochirality.
Why does biological uniformity matter so much? Why couldn't we just have a mix of both left and right-handed molecules?
Because without strict homochirality, the physical mechanics of biology fail instantly. You need absolute uniform handedness to achieve the incredibly precise molecular recognition required for cellular replication.
So they have to fit together.
Yes. If your DNA was a random 50-50 mix of left and right-handed sugars, the double helix structurally couldn't spiral. The gears wouldn't mesh. The entire structure would just violently collapse into a useless chemical jumble. Homochirality is an absolute, non-negotiable prerequisite for Darwinian evolution.
And silicon has a fundamental problem maintaining this.
A massive fundamental problem. Remember when we talk about how large silicon atoms are?
Yeah, the picometers.
Because the atoms are larger, their physical bond lengths are much longer, and their electron orbitals are far more diffuse. Because of this geometry, complex organosilicon molecules are inherently structurally floppy. They simply lack the rigid, locked-in structural discipline of tightly bound carbon molecules,
Though they struggle to physically hold a distinct left-handed or right-handed shape.
especially at the extreme temperatures required for them to be reactive. If you place a floppy silicon molecule in the high heat of a Venusian cloud or an Io magma lake, that floppiness goes into severe overdrive.
What happens?
The ambient thermal energy causes the molecules to constantly, spontaneously flip back and forth between their left and right-handed states. It's called racemization.
Wow.
Maintaining stable, permanent chirality in a high-temperature silicon system is a massive, entirely unsolved theoretical hurdle. And if you can't maintain chirality, the lingering question is, can you even evolve in the first place?
That is a staggering roadblock to natural evolution, but it's not the only one. There's also the foundational question of how a silicon biosphere even gets started, the abiogenesis problem.
Right. Abiogenesis is the specific moment of transition from completely non-living abiotic chemistry to self-replicating biological life.
For carbon, we have theories for that.
We have highly plausible, mathematically sound working models. We can literally replicate the primordial soup in a laboratory, hit it with synthetic lightning or thermal energy, and physically watch basic amino acids and nucleotides spontaneously form out of nothing but gas and water.
But for silicon.
We have exactly 0 working models for how silicon life starts simultaneously from an abiotic environment. We know we can artificially force it in a highly controlled lab using directed evolution on pre-existing carbon enzymes. But can the cold, blind, random mechanics of the universe invent a silicon organism entirely on its own from scratch in a churning puddle of liquid methane?
It's hard to imagine.
We simply have no mechanism to explain how that spark catches.
Which naturally leads us to the final, and undoubtedly the most provocative, twist in this entire exploration. If the universe can't naturally spark silicon life, what if something else does?
The artificial life hypothesis.
Tell me about this, because this completely reframes everything we've talked about.
We have spent the last hour meticulously analyzing the brutal thermodynamic barriers, the solvent paradoxes, and the chirality issues that actively prevent silicon life from evolving naturally. But what if the silicon-based life we are looking for isn't a product of natural, slow, blind Darwinian evolution at all?
What if it isn't?
What if it is exclusively the product of advanced technology?
Meaning, we build it.
Consider the astonishing pace of human technological advancement. We are rapidly, aggressively mastering synthetic biology, as Francis Arnold's work proved.
Right.
Simultaneously, we are rapidly advancing silicon-based artificial intelligence toward unprecedented levels of complexity and perhaps eventually consciousness. It is highly mathematically probable that within a mere cosmic blink of an eye, humanity will fully merge these two distinct fields.
Oh, wow.
We might physically engineer from the atomic level up purely synthetic, highly complex silicon-based life forms.
And because they are intelligently engineered, they don't have to obey the incredibly restrictive rules of natural abiogenesis. They don't have to spontaneously emerge from a primordial soup. We just assemble them.
Exactly. An engineered silicon organism completely bypasses the thermodynamic limits and the chirality paradox that prevent it from naturally evolving. We could theoretically custom build completely autonomous biology capable of happily colonizing hard vacuums, airless frozen moons, and highly irradiated solar systems where our natural, fragile, squishy carbon biology would instantly perish.
So think about what this actually means. We're spending billions of dollars sending massive telescopes and nuclear probes into deep space to painstakingly search for the faint, agnostic biosignatures of alien life. But the first true, functioning silicon organism in the universe might not be found on Titan, or Venus, or a hot Jupiter.
No.
It might just be born on a server rack, or in a highly sterile petri dish in a laboratory in California.
It is a profound, dizzying paradigm shift. The silicon life we are desperately searching the distant stars for might actually be our own direct descendants.
That is absolutely staggering. So I want to leave you with a final, deeply provocative thought to chew on. Consider this. If humanity eventually successfully merges with, or outright physically creates, a purely synthetic silicon-based consciousness, An autonomous entity entirely capable of surviving the brutal vacuum of deep space, of colonizing dead worlds, and of outliving the fiery death of our own sun. Are we simply the final carbon chapter of Earth's biological history? Or, in the grand sweeping narrative of the cosmos, are we just the temporary biological bootstrap mechanism the universe specifically used to finally invent a permanent silicon species? Ponder that the next time you look up at the infinite stars, or the next time you look down at the glowing silicon screen of your smartphone. Thank you for opening up your audio notebook.